My name is Dr. OWUNA, the course lecturer of MCB111. I'm here to introduce you to general microbiology, the following course content: definition of microbiology, history of microbiology, principle of microscopy, types of microscopes. By definitions, microbiology as a phrase was derived from the Greek words meaning micros as small, bios life, and logos as science. And then when you put this together, you have microbiology as the study of microorganisms which are too minute to be seen clearly with our naked eye. Microbes, also called microorganisms, is a term applied to all microscopic living organisms, that is, the minute living organisms. And these groups of organisms include bacteria, fungi, yeast, molds, protozoa, algae, and viruses. Those are non-cellular entities, sometimes regarded as a straddling border between life and non-life. The study of microbial cells and how they work and affect man, plants, animals, and the environments. The history of microbiology. The field of microbiology developed further and gained its importance after the fascinating discovery around the 16th centuries by the discovering of microscopes by pioneer scientists. The study of microbiology will always be incomplete without a touch on the founding fathers of this important field who have contributed significantly to the development of microbiology. It is important to note that several scientists, microbiologists in the time past, have contributed tremendously to the development of microbiology, and the notable amongst them are Robert Koch, Robert Hooke, Anthony van Leeuwenhoek, Lazarus Spallanzani, Louis Pasteur, and a lot of them. The contributions of Robert Hooke, who was the first person to discover the cell from the cross-section of a cork. He noticed some microscopic fungi too. He also developed a simple microscope of 30 magnifications and observed few microorganisms. Anthony van Leeuwenhoek, who is usually and widely regarded as a father of the field of microbiology. He was a Dutch merchant and a haberdasher, and his hobby was to make lenses and microscopes. His microscopes were simple microscopes composed of double convex glass lenses held between two silver plates that could magnify 50 to 300 times. He was the first to describe the bacteria and protozoas, which is so, and he described them as animalcules. The theory of spontaneous generations, also known as abiogenesis. After the discovery of microorganisms by Anthony van Leeuwenhoek, scientists began investigation about origin of microbes. Before then, the origin of microbes was oblivious and was not well known. But after the discovery of microorganisms by Anthony van Leeuwenhoek, scientists began investigations about the origin of microbes. Since organic matter decomposes quickly outside the living body, it was assumed that microorganisms were arising by spontaneous generations, which was further supported by Francesco Redi, who supported the theory of spontaneous generations. He boiled the meat and covered the mouth of the flask with a wire gauze. The flies, attracted due to the odor of the meat, laid eggs on the wire gauze that later developed into maggot. Thus, he established that origin of maggot was from meat and not from flies. Additionally, John Tender also contributed, who is an Irish priest. He observed the appearance of microorganisms in a putrefying meat and interpreted it as spontaneous generations. Some of these theories were not supported or agreed by some other scientists. However, Lazzaro Spallanzani in 1729 to 1799, he brought his own theory of biogenesis just to counter the theory of spontaneous generations of abiogenesis. Spallanzani is an Italian priest who boiled beef broth for an hour. He sealed the flask and observed no appearance of microorganisms, and disproved the theory of spontaneous generations or abiotic origin of life, and proposed the theory of biogenesis. He said that every form of life takes its origin from their parents, germ cells or seeds. And this theory of biogenesis was later proved and supported by Louis Pasteur. And then Louis Pasteur, who was a professor of chemistry at the University of Lille, France, he is considered as pioneer of microbiology, as his contribution led to the development of microbiology as a separate scientific discipline. He proved the theory of biogenesis and disproved the theory of spontaneous generation, abiogenesis. His theory went contrary to abiogenesis, which is the theory of spontaneous generation. He experimented it by using a swan neck flask, whom he passed the untreated and unfiltered air into the boiled nutrient broth. Germs settled in the gauze neck, and no microbes appeared in the solution. And thus, he disproved that living organisms appear from non-living matter. He also worked on souring of wine and beer and found that this alcohol spoilage is due to the growth of undesirable organisms, such as bacteria, fungi, and other things that can ferment and cause undesirable odors. While the desirable microorganisms produce alcohol by a chemical process called fermentations, he showed that wine did not spoil. It is if it is heated to 50 to 60 degrees Celsius for a few minutes, the method which he later described as pasteurizations, and now is widely used in dairy industry to kill pathogenic microorganisms in milk. He is a founder of germ theory of disease He visualized that diseases are caused by microorganisms. In course of his research, he discovered that the importance of sterilization and discovered steam sterilizer, autoclave, and hot air oven. He also contributed in the importance of cotton wool plugs for protection of cultural media for area contamination, so that to prevent the contamination that may come from the air. He differentiated between the aerobic and anaerobic bacteria and coined them the term as anaerobic, which he later referred to the organisms that do not require oxygen. That is, organisms that do not require oxygen for growth are usually termed anaerobic organisms. He worked on pembrine, a silkworm disease caused by a protozoa, and showed that infections can be controlled by choosing worms free from the parasite for breeding. He developed the process of attenuations. And attenuation is just like is a term that usually is referred to reducing the pathogenicity of a disease causing agent. During his work on chicken cholera in fowls, he found that cultures which had been stored in the laboratory for some time would not kill the animals as fresh cultures did. This attenuation is now used in protective vaccination against disease. Then we have John Tyndall, who also designed a special chamber to free the dust in air and kept the sterile broth in the chamber. And he discovered that no microbial growth was observed when sterilized broth was kept in the chamber. Thus, he proved that dust in the air carried germs, and this is a source for the growth of microorganisms, not the spontaneous generation. He also developed a sterilization method called Tyndallization. And Tyndallization is otherwise called as the intermittent or fractional sterilization. In a case of Tyndallization, subsequent heating and cooling by steam for 3 days usually removes the germs and their spores. And heating at 100 degrees Celsius kills the vegetative cells. The spore cell forms are also killed on subsequent heating upon germinations. Robert Koch. Robert Koch is one of the scientists who has contributed immensely to the development of microbiology. He is a German country doctor who later became the professor of hygiene and director of Institute of Infectious Diseases at Berlin. He performed several bacteriological techniques and is known as the father of practical bacteriology. He discovered the rod-shaped bacteria, rod-shaped organisms in the blood of animals that died of anthrax. He experimentally obtained the anthrax organisms in a pure culture on a depression slide by inoculation of infected blood into the aqueous humor of a bullock eyes. He observed multiplication of bacteria and spore formations. He injected these spores into mice and just to see that these spores reproduce the same symptoms into these mice. He found that in a certain conditions, the anthrax bacillus forms spores that can survive on earth for years. He actually passed the anthrax bacilli from the blood of an infected animals from one mouse to another through 20 generations and found that they bred through. He worked out its life history. He brought the staining techniques. He prepared a dried bacterial films, smeared on a glass slide, and stained them with the aniline dyes for producing so as to produce a better contrast under microscopes. He discovered tubercle bacillus, a Mycobacterium tuberculosis, which is popularly called as Koch bacillus. He then injected these tubercle bacilli into a laboratory animals so as to reproduce the disease. Satisfying all Koch postulates, he discovered Vibrio cholerae, the causative agent of cholera disease. He went further with his experiment to develop a pure culture techniques by introducing solid media, the use of agar obtained from dry seaweeds. In the preparation of solid bacteriological media, it was first suggested by Frau Hughes, the wife of Koch's student. This agar is totally inert with no nutritive value. However, it solidifies at 45 and also melt at 90 degrees Celsius, and was found to be most suitable solidifying agent in the preparation of some culture media. Koch also went further to isolate bacteria in pure cultures on this solid media, which he revolutionized the bacteriology. He also discovered that old tuberculin. He also noted that when tubercle bacilli or its protein extract was injected into a guinea pig already infected with the bacillus, and exaggerated reactions took place, and the reactions remains localized, and this is particularly called as Koch phenomenon, and it is a demonstration of cell-mediated immunity. Tuberculin test is based on Koch phenomenon. He erroneously thought that protein extracted from tobacco bacilli called old tuberculin could be used in the treatment of tuberculosis. Koch did a series of experiments to ascertain the veracity of the criteria laid by his teacher, Hen, and then to establish the causative role between a particular diseases and a particular disease, a particular microorganisms, which later known as Koch postulate. He postulated that for a particular disease to be associated with the particular microorganisms, a specific organisms should be found constantly in association with the disease. 2, that the organisms should be isolated from the disease-causing organisms, should be isolated from the associated organisms that was specifically identified as a test target where the disease is associated, and that the organisms should be isolated and grow in a pure culture in a laboratory, and that pure culture, when inoculated into a healthy animals or organisms, a susceptible organisms, should be able to produce the similar symptoms as the same as the legions of the same diseases. And then from there, the inoculated animals should also be isolated from the microorganisms, should also be isolated from the inoculated animals. And additionally, the specific antibodies to the causative organism should also show demonstrable symptoms in a patient serum. The next father of microbiology who has contributed a lot is Edward Jenner. He's an English country physician who discovered a safe and efficient vaccination against smallpox, which ultimately led to the eradication of smallpox. He was finally known and called the father of vaccination or immunizations. Jenner observed that dairy workers exposed to occupational cowpox infections were immune to smallpox. He proved to them experimentally that resistance to smallpox can be induced by injecting cowpox material. The next one is Joseph Lister, who is popularly known as the father of antiseptic surgery. Joseph Lister was a professor of surgery at the University of Glasgow and Edinburgh, and he later at the King's College, London. His interest is deeply in the prevention of post-operative sepsis. He was attracted by Pasteur's germ theory of diseases and concluded that sepsis or wound infections may be due to microbial growth. That is, once you have a sepsis or wound, it is as a result that perhaps microorganisms must have gotten in contact with the wound. He successfully prevented post-operative sepsis by introducing antiseptic techniques. Lister's antiseptic surgery later led to the development of aseptic surgery. And the next father of microbiology is the Ivanowski. Ivanowski, who is a Russian botanist, occupies a pivotal positions in the history of virology. We also have a Dutch microbiologist, Benjarikai, showed that infectious agent could diffuse through an agar-wool gel, and that it was a non-capsular contagious vivium fluidus, which he called viruses. Many scientists or microbiologists made some contributions to the development of microbiology. For instance, the Alexander Fleming, who was an English scientist, and he brought about lysosome and penicillin, the discovery of lysosomes and penicillin. He discovered that lysosomes, by demonstrating that the nasal secretions has the power of dissolving or lysing certain kinds of bacteria, thereby rendering it ineffective. He demonstrated that lysosomes was present in many tissues of the bodies, which has an enzymes that neutralizes some infectious pathogens. Then this moves us to the second topic or title or content, the principle of microscopy. We all know that scientists have proved or demonstrated to us that microbiology is the study of microorganisms that cannot be seen with our naked eye, except with the use of microscopes. And we're meant to understand that the first scientist that discovered the microscope, who was Anthony van Leeuwenhoek, he first of all described these organisms, who he names them as animalcules. He said these organisms were just wriggling the ponds of water, and he described them as animalcules. And these organisms is a Dutchman, a haberdasher and a lens maker. He described these organisms as animalcules. We have all kinds of microscopes that use visible light to observe specimens, and a number of them have some characteristic features that differentiate themselves. We have the simple microscopes, the light microscopes, and the light microscope is part of the wide spectrum of electromagnetic radiations from the source. It is a form of energies that most light wavelengths reflect the length of light ray, and it produces a continuous spectrum of electromagnetic radiations with waves of various lengths. Lenses are optical devices which focuses or disperse a light beam by means of refractions. It is a simple lens consisting of a single piece of transparent material, and its light rays from a distant source are focused at the focal point. It has a numerical aperture, which is NA, as the value representing the light gathering capacity of an objective lens. The numerical aperture was first described by Ernest Abel and is defined by the following expressions. Numerical aperture, which is NA, is equals to N times sine Q, and the N is the refractive index of the medium between the specimen and objectives. And then the theta is a half aperture angle or collections angles. We have the types of microscopes. In order to view microorganisms or microbial cultures or structures of different sizes, it is important that different kinds of microscopes are used. The light microscopes resolves images with the help of light, and the specimen is usually viewed as dark object against a light background in bright field microscope. And the dark field microscope uses a special condensers, and the specimen appears light against a black background. The other types of microscopes are phase contrast and fluorescence microscope. We also have an electron microscope, which usually uses a beam of light which usually uses a beam of electrons instead of light. An electron passes through the specimens and form a two-dimensional image in a transmission electron microscopes. The principle of microscopes. Each microscope has its own principles. The modus operandi of a microscope differs. For instance, in a simple microscope, light microscope, it has its own principles. For instance, a microscope enlarges the view of an object by enlarging it twice using the objectives and oculus lenses. And it happens in every following ways. 1. The light from below the specimen illuminates it. 2. An objective lens placed very close to the specimen produces a real image of the specimen. 3. This image is created somewhere between the two lenses. The oculus lens looks at the real image already magnified and create a virtual image of it that is small, that is many times magnified. This image is created, the images are created far away behind the actual objectives. And you can see the diagram of a working principle of microscopes. where it has the eye pupil, the eyepiece, the optical length, the objective lens, where you can adjust the focal length. This is where you will now view it from your eyes to magnify it. This is the focal length, and then the focal length also has an optical tube holding it firm. And with this we have come to the end of this lectures in the first the content, the use of microscopy, introduction to general microbiology, the contributions of scientists. So in our next we'll go further to treat other topics or content as we go further. Thank you very much.